Let's talk about panarthropods, which is arthropods and all the things that are pretty close to arthropods, but are not quite arthropods. So these would all be in the group called the ecdysozoans, which are things that have a coating that they are going to shed as they get larger. That's definitely true for arthropods, but there's also some soft bodied creatures and creatures that fall outside the arthropods that are like this. So let's get into it. Yeah. All right. We'll start as we do every day, back in the Ediacaran. And in the Ediacaran, there are some creatures that are surprisingly close to being bilaterally symmetrical. There's one little worm that is bilaterian that you find in Ediacara fossils. But then you've got things like this, Archispinus. So this is from the Ediacaran, and what is it? You look at that at first, you'd say, "Oh, that's like it kind of looks like a trilobite. It looks like it's got a head. Looks like it's got a tail. Looks like it's curving a little bit to one side." But then you remember, this is the Ediacaran. We have things like Andiva. We have things like Dickinsonia. And they also have what appear to be little segments, but they are more like a charnia, more like a rangiomorph. You know, those things that are like animal ferns than they are like arthropods in terms of how they grow. They're sort of constantly adding segments as they grow, which is not usually a thing with arthropods. They'll develop up to a point and then they keep shedding their exoskeleton in order to get larger. But they don't just arbitrarily add segments forever and rearrange their body plans the way these do. So, yeah, some people have said, well, maybe this is in the crown group. And honestly, it's probably in the crown group for something, but I'm certainly not going to be the one to go out on a limb and say exactly what, because these are weird. And other than having animal like cholesterol, I think that's the only thing we can say for sure about this group. So, when do arthropods get going? Well, you really only find them from kind of not the earliest Cambrian, but from about 521 million years ago. That's when you get the first trilobite fossils that are unambiguously identified. But let's get into it because there's more to this. All right, so it's from a nice PNAS paper, and you can open that up, put that in your browser. It'll take you to the paper itself. Here's our timeline. So you've got your Ediacaran with all these crazy Ediacaran creatures, and that's what all the blue dots are. These are things other than arthropods. And then you've got sites that have arthropods. So the first appearance date is what the FAD is. This at 521 is the earliest actual fossil we have of trilobites. And the weird thing is that you get these first fossils in Siberia, and you get them in Morocco, and you get them in other parts of the world, which are and were, because that's important to think about, spread out at the time. So these are a global phenomenon when we first start noticing them. And that's a problem because, yeah, nobody starts as a global phenomenon. You got to earn it, you got to build up over some amount of time. Unfortunately, that time was before the first Burgess Shale type deposits, where we get all this evidence for all the soft bodied things in addition to the hard bodied things. And so most of what we have between 521 and 540, 541, wherever you draw the line these days, is little parts in small shelly fauna. And some of these could be arthropods, and some of these are almost certainly not arthropods. And it is very difficult to determine which is which. But you have plenty of other groups that are developing here. You have the earliest mollusks. You have earliest skeletonization, things like those little cup in a cup fossils, Claudinia being one of those. Yeah, and so there are certainly armored animals before there are arthropods proper, with arthropod type internal organs in addition to the exoskeleton. It's not just all about the exoskeleton. Yeah. Here are some very early ones. So these are from Orsten. I'm probably mispronouncing that, but it's ORSTEN type preservation. This is found in some locations. It is a phosphatic preservation, so you get phosphate minerals combining with a lot of the different organics on there, and that ends up preserving them in a way that you can dissolve the rest of this little ball that is formed around them, little ball of mud. That goes away, and then you sort through it, and you'll find pieces of exquisite little phosphatized fossils like these. And they're covered in legs. And really, the only way you can be covered in legs like this, we're looking at exoskeleton, is if these are somewhere at least on the way toward being arthropods. And so the group are Euarthropoda, like the true arthropods, but then there's also Panarthropoda, which are arthropods and friends. They're the wider world of arthropod-like things. So these things exist, and in the same formations, you can actually find what look like modified, somewhat recognizable versions of some of the feeding appendages, I believe, of certain types of arthropods. Shown in E, this is currently the oldest unambiguous euarthropod. It's actually a crustacean, Eucharis, also preserved in one of these little dissolvable fossils. But earlier than you get the first arthropod fossils, you start to see the first probable arthropod traces. So things like Rusophycus, which remember is the little resting trace, where trilobites have used their bilaterally symmetric appendages to dig a little spot to rest for the day, the night, or just because. Yeah, we don't know. And so those actually date back even earlier. Here's one of the first trilobites, Phylotaspid trilobite. Phylotaspis being one of the very earliest ones. Trilobites are some of the first arthropods or panarthropods to get going. Yeah, and they're a little different than some of the others, but they certainly achieved worldwide distribution earlier than any other group that we know of for certain. I think eventually maybe something will push them off of that pedestal, but for now, that's the way it looks. They got big early. It's kind of like a founder effect, like how the first company to enter a economic space tends to dominate that space and just get huge, and then it's very hard to displace them. Yeah, looking at you. Google wasn't first, but boy, did they do it better. Yeah, yeah, that's the idea. So here are some panarthropods from the Burgess Shale. And so this is later. Burgess Shale is going to be something around 514, I think. Remember, the earliest trilobites were at about 521 million years ago. And so these panarthropods have diversified, and you have all kinds of crazy things. You have Anomalocaris with the two appendages at the front, with the sort of scissoring, radially closing, irising mouth parts where they all just come together, we think, which is actually a very old ancestral panarthropod trait, but we'll get into that. And all these other crazy swimming things. That's an Opabinia, I believe, in panel B. That's the one that has 5 little eyes on stalks on top of its head. Because why not? It's the Cambrian, anything goes. The rules were not yet set down, or rather the possibilities were still greater. Slight tweaks to developmental patterning genes seemed to work okay at this time, and led to what looked like some really funky, very different types of animals, which I suspect if we ever were able to come up with some proxy for DNA sequencing, I bet these things wouldn't be that far apart. I bet they're all right down there in the crown from which the different types of arthropods spawn. But yeah, remains to be seen, as just a guess. Some of our favorite arthropod adjacent organisms, panarthropods, are going to be lobopodians, Lobopodia. And these are going to walk along on little legs. They have little rows of sort of curly legs. They have little hooks at the end of these, like little toenails. And they have a long body, which contains a gut. Quite often, they're going to have some form of protection, which is going to be on the top of that, kind of a "don't eat me" thing. A little notice, perhaps, to trilobites and anomalocarids and whoever else was snacking on other invertebrates at that time. So this one called hallucigenia, because apparently people thought they were hallucinating when they saw it because it was just so weird, which I get. This is what you got. So sitting down here at the bottom, barely marked, these little tiny threads, those are the legs. And each of these legs has little tiny claws on the end of it. So these are the appendages, and the boxes are going to be around some of these claws. And then at one end, they have a sort of a bulbous protrusion. This turns out to be the head. The original diagrams of this had it at the opposite end, or people just said, we don't know what this is, or people had this upside down, originally reconstructed, with the spikes holding it in the ground, like a brachiopod. I like that people were thinking like brachiopods. That's very good, very encouraging for the world in general. Yeah, and basically nobody knew what was going on with these things. So let's talk about them a little more. A recent paper that you can get if you right-click on some of these blue letters here, those hot links would still be valid and will take you right into the paper. These turn out, now that they've looked at several of them and looked very closely and used a little bit of molecular paleontology and just sort of better resolution, what you can see is that zooming in on this end, which is the little blob that we now think is the head, there are two little dark patches there. Look at that. What has two little dark patches right on its face? Well, that'd be anything with eyes, right? Here we're showing it under a different form of illumination, if you like, a different imaging method that really picks out the eyes. And it turns out it's not just one of these, it's all of them. You can always see the two little spots there. On this one, part of it broke off as the rock is being separated. It doesn't always part exactly where you want it to part. Yeah. But yeah, it would appear that they had at least eye spots and probably something like eyes. So that's cool. And then what's this little Cheshire Cat smile we've got here? Here it is turned sideways. And here it is diagrammed. This looks like, and probably is, the circumoral, so around the mouth, elements. And these circumoral elements are basically like teeth that are in the shape of a circle. So it's a little circular mouth that can presumably close from all directions and really munch and crunch whatever goes in there. These had a mouth like an anomalocaris. And anomalocaris is thought of, or at least was originally thought of, as this super fierce predator, which it may still be. It was one of the largest, most mobile organisms found in the Burgess Shale and Chengjiang. But these guys are actually built fairly similarly. Yeah. And then, because why not? So let's take a step further down. You can see lining this little track right here, these little tiny dots, and here they are all, look at that. These little tiny dots. These are little mineralized structures that would be down in the throat, basically. So these are little teeth that will help with either further grinding whatever gets pulled into the mouth of this now hideous-looking hallucigenia beast, or they will prevent anything that goes down from coming back up because they're going to be pointed in this direction. So swimming the wrong way will essentially get you impaled. So yeah, we've got a set of organisms that have a common sort of feeding mode, these sort of rings of teeth, and then tooth-like elements down the throat, which are called aciculi. Aciculi right there. And these are right down at the base of the Panarthropoda. So that's a feature we can look for down there. All right, so here is just the coolest reconstruction. This guy of a Lobopodian. Look at that. It's hallucigenia, I think. Looking fantastic. This one has all these little sort of elements up near the mouth, possibly additional feeding parts of some sort or harvesting food, one would imagine. And those, yeah, sort of spines coming out of the back, little rigid spines and little rigid claws down here. They're not necessarily heavily mineralized, and it's real hard to tell because this is Burgess Shale, so all the hard parts are gone, and we have casts of them essentially. But they did fossilize really well, and they did fossilize in a way that tells you they were rigid. They don't get smashed flat. They retain some of their three-dimensionality, which means they had at least some sort of heft to them. They're able to resist the crushing. Yeah. But yeah, here is the hallucigenia, and it's rearing up to come and get you. These things were about this big, most of them. So you don't have to be too worried tonight. But yeah, you got your right and left eyes. You got your buccal apparatus up here. You got your ring of teeth that go around the mouth, and the circumoral elements, and you got your isiculi, which are the little teeth that are going to be all down the throat, which could presumably, if it's a muscular throat, help to mash up whatever that was that these are eating. So they look like either little tiny grazers or little tiny predators. I guess it's kind of hard to tell. But this is a apparatus that could be adapted to either of those uses. So here are, this is one of the first fossils ever published of hallucigenia, and these are Lobopodia for sure. These are some also Lobopodia. So this one's called the walking cactus Diania chengjiangensis from Xinjiang. And look at this thing. It's a Lobopodian, so it has these little feet. Each of the feet has claws on these presumably soft appendages. They're very bendy. They're always at different angles. They are sort of built in serial segments like you would maybe get in a worm, so maybe some affinity there, but also like you would get in an arthropod. And look at our trilobite up there. Lots of serialized appendages there. Yeah, so a common thing that we would expect to find perhaps in the crown group there. And you can see the little head here, and I don't know that anybody has looked for the eyes or whether they would even have been preserved at Xinjiang. I think it is just the melanin at the back of the eyes that ended up getting preserved. I don't know why that didn't dissolve in the Burgess Shale fossils, but it made it, and I am glad it did. Yeah, certainly glad. Here is also a Lobopodian. Now, this is Microdictyon, and they're really tiny. These are interesting because they have these little plates on every segment. So they don't have like the overall spiky body like this. They don't have the gigantic spines that are as long or longer than the legs. But they have these little shields all on the side, like, I don't know, drawings of Viking ships you might have seen as a kid. And the shields have an interesting structure. They're sort of hollow, and they've got these little sort of, it looks like little marbles stuck in there. There is some suggestion that these may be a visual apparatus, maybe sort of secondary visual apparatus. You'd have your eyes up front. And on a lot of things like brittle stars, for example, there are sensory tissues throughout much of the body, so they can really see you through their armor. And it's not at all certain that that's what this is. But also, if you've got the ability to form something that looks like a compound eye and serves the purpose of defense, and you happen to have a little tweak that puts the genes for that thing, they start growing it over your regular eye, do you not have something like a compound eye? Potentially. That's what people were thinking, or those are the general lines, rather unsophisticated presentation of them. Here is a later Lobopodian. So these things survived for a while. They're just small and very soft bodied. So you only get them in places where you get soft body preservation. These are from those famous sites, that one in the Silurian, where they'll section the creature and photograph it at each level and reconstruct it just like that. And here you can see, like, all the legs on this side are pointed up and all the legs on the other side are pointed down. I think that's great. That's the position in which it appears to have died. Yeah, and so you've got some detail up there around the head, the sort of buccal area, and of the overall organization in 3 dimensions, which is pretty cool. Yeah. But who else has these adaptations? Well, there are a group of arthropods called pentastomids. And pentastomids look just like a facehugger from Aliens. They're sort of legs up here and a body, and then a long tail that hangs on the bottom. Just like that. Sorry to frighten you. But if you compare, there are Cambrian pentastomids that have been found in this Orston presentation. And their appendages look very similar. They've got a little sort of zipper right here, which I think may be where it splits in order to molt. And they've got little claws on the ends, just like you find little claws on the tips of these modern ones. So pentastomids, showing something in common with lobopodians. Yeah. Who else has stuff like that? Why, tardigrades, everybody's favorite little microscopic, not quite arthropod. So tardigrades are in their own phylum, Tardigrada, and so they are next to the arthropods, and they would be in the Panarthropods, but not in the Euarthropods. They're held aside. They're going to have a leathery casing, so they're ecdysozoan. They're going to shed their skin, basically skin. Yeah, it's not really like our skin, but they're going to shed it in order to grow. And comparing modern tardigrades to the Cambrian one that we found, I think it's still just one. You see the biggest difference is that all the parts up here are basically the same. It's just that the modern one has grown 2 little limbs, additional limbs, out of this end, out of the distal end of its body. Not a gigantic change as they go. Yeah. And here are some of the other panarthropods that you find in this Orston fossilization. This one has been considered to be a type, an agnostid, which were originally considered to be definitely trilobites, but maybe trilobitomorph, like trilobite-like features, and close to the trilobites, but not actually trilobites. That's an area of ongoing discussion, but that's where it is settled at the moment, and I don't know, talk to me in a year. We'll see how we go. Here, this one I think is a pentastomid up here. Yeah, looking great. Looking like a little alien. And you got things that are basically going to have a bivalved carapace and a little shrimp-like organism in there. So similar style to what you would expect for an ostracod, but the arrangements of all the parts are different. So it is not. Yeah. And it's pretty amazing that you can get these little tiny, they're only fossils of usually larval things, but that you can get this preservation is just marvelous. All right. So, yeah, all right. We've got those other things like tardigrades, for example. But what about their mouths? You're asking. What do their mouths have a little ring? Do their mouths have little teeth and stuff? I say, just wait a minute. Just wait a minute. Because look at the little claws that you get on the tardigrade, and then see how the base sort of fans out like this? That's actually what appears to happen in the lobopodian claws that you get down here. They widen out at the base and they sort of fan out. They have sort of multiple projections, and then on top of it, they have this sort of little hooked claw. So the claws are rather tardigrade-like on these lobopodians. Are lobopodians tardigrades? No, they're in the crown group. They're their own thing, but yeah, they're not far off. They're showing a lot of the same features, a lot of the same traits. And here we're looking at the buccal apparatus of tardigrades, and sure enough, they have a little ring-like apparatus down here. And then this area right down here, it's essentially muscular pharyngeal apparatus. This is going to hold food in this little tube-like chamber and just sort of mash it to grind it up for ease of digestion, which is exactly what those little teeth down in the throat of hallucigenia presumably would have done. It would have had a sort of a longer buccal apparatus, but that doesn't hurt a thing. That's just, a bigger conveyor belt for more chopping, more fine chopping. Here is the oral, the circumoral teeth that you get on, they're not really teeth, they're not made of dentine, but on tardigrades. Here's what you get in some of these fossils. And so you can see right there, those are very similar. These appear to be trying to do the same thing. So yeah, it looks as though this is a very early arthropod trait, and not surprisingly, we see it in a variety of very early arthropods, which may have been genetically fairly close to each other, just sort of differences in which developmental genes are turned on at which time, sort of in a time before those were really as rigidly set. And I think developmental genes only have to be rigidly set if you have further development that is happening after those are patterned. So like in humans, we grow limb buds, like these little sort of chicken nugget looking things, and then we fill them with bones. We develop vasculature inside them. Then we grow additional digits at the ends. And so each thing is sort of stacked on a more primitive adaptation. But if you're down near the base, if you're down in that stem group, in that crown group, it is possible that there were more possibilities, or it was easier to access those possibilities from there. So here's how Panarthropoda looks according to that same paper in PNAS. So you've got your things like hallucigenia. Those are panarthropod, but those are kind of outside of the bounds, and they're really early. These are very close to what we think is the dawn of actual evidence for arthropods, at least for now, until somebody finds something a little bit older. But they weren't around in Ediacaran, because we've got good soft body preservation then. And they're definitely the most abundant things by the time you get to the middle of the Cambrian, in the 15 to 520 million years ago range. And so it's just a question of how fast they get going and when we can detect the first steps of the inevitable march of the arthropods through history. Yeah, they're doing great. Don't worry about them. Yeah. So yeah, there's hallucigenia on its own little branch up there. Then there's a question of whether these onychophora, so these are the velvet worms that some people keep as pets. They're awesome little kind of soft-bodied predators. They are not arthropods, but they're not far from being arthropods. They're in the panarthropods. So are they a branch from really early, or are they a branch from up here, which they would share a lot more traits with euarthropods. And here are our first euarthropods. But look who's right before that. You get your radiodonts, which are things like Anomalocaris. That's the two big sort of grasping appendages there. The stalk eyes for maximum hunting prowess. The nice little modified sort of soft but covered, they kind of work like wings. It's modified body parts. Yeah. And great little sort of predator setup. Over on this branch, you're going to have some of these early things that are somewhere between tardigrades and other panarthropods like Lobopodia. And so this is one of them, and that's what this paper was about. And then, you'll have your split where all the tardigrades fit here. And those can then be broken down into different groups. And since tardigrades are still around, we have genetics for those, and genetics for these, and genetics for onychophorans, which means that kind of we have a lot of genetics that could define what the original set of components that early arthropods were using would have contained. All right, that's enough. with panarthropods, we'll have separate things on, for example, trilobites coming up. And I know you're excited about that too. All right. Thanks a lot.